So very happy to be here and to present my work. So I'm a second-year PhD student at Institut Imagine, and today I wanted to discuss with you about iPS-derived motor neurons for modeling ALS, and more specifically how we do drug screening in this model. So iPS-derived motor neurons, first time we saw it in ALS was 2008, where we were able to show that we can get iPS from ALS patients and differentiate them into motor neurons. And so that was a big deal, because since then there have been, don't worry, I'm not going to put you here, the more than 500 papers that have done ALS modeling on iPS-derived motor neurons, more than 50 different reviews. But this has led to the Japanese team that did drug screening on this iPS-derived motor neurons and detected a potential therapeutic agent that was later brought to a phase 1/2 clinical trial, so it was ropinirole. So we have, in the ALS field, used this model. We are doing drug screening, but what we had as a question was, what parameters are we currently using to do drug screening in this motor neuron? So what are we rescuing in this motor neurons as a parameter? So in the lab, we work mostly on TARDBP, which by now you know encodes for the TDP43 protein that is mislocalized in patients in ALS patients. So we asked the question of, does TDP43 mislocalize in iPS-derived motor neurons? Because today this is one of the parameters that we use to do drug screening. And so I checked all the papers that have looked at TARDBP patients and checked if they see mislocalization. And well, the answer is yes and no. So in between studies, sometimes it's yes, sometimes it's no. We try to link this with mutations, with what coating that they use, what age they looked at the motor neurons. And we didn't really find a link between that. And then we checked the quantification techniques, and sometimes the same technique in different labs leads to different results. So maybe this is not a really reliable parameter for drug screening in iPS-derived motor neurons. So we asked, how can we improve drug screening in this model? And specifically here, we focused only on TARDBP cells, just to kind of simplify a bit the process. And for that, we wanted to develop a screening assay that is high throughput. This way we can try a bunch of different drugs and also do it in different cell lines. We wanted something that was automated in order to get out all the bias that we can add when we were experimenting. And lastly, we wanted a reliable parameter, not as the one that we saw. So in the lab, we have different iPS lines. We have ours from the Cedars-Sinai Hospital. So we have 4 TDP lines directly from patient, and we also have the COLF line with 2 TARDBP mutations included inside. But today I'm not going to present results on these COLF lines. I know this is the line that is being used a lot today in the field, but sometimes we see differences between our lines, our patient lines, and this line. So I'll be happy to discuss about this later. So today we're only going to discuss about the patient's lines. So in the lab we use the Maury et al. Protocol that differentiates the iPS from embryonic bodies. And after this protocol, we're able to get motor neurons at around 85 to 90% of islet positive cells, so pretty good ratios, and in a similar efficacy between different cell lines. So we used these cell lines, and first we asked the same question: does TDP mislocalize in TARDBP iPS-derived motor neurons? And we did this. We tried to automate the process to see if we automate the process, would we see anything? So for that, we used the Opera Phoenix high content screen, which is an automated confocal microscope. So we just put plates, 96 well plates in there, and then we get images on all of the wells. And so we, again, automated the process of finding the nucleus, finding the cytoplasm. This way we're not touching in the analysis process. And here you have TDP, very nuclear, but we checked specifically the cytoplasmic versus nuclear ratio. So in green you have our control lines, and in red the TDP lines. And so you see nothing really striking. When we pull all together, we see a slightly increased TDP43 expression, so more in the cytoplasm, in the TDP lines, but nothing striking. And maybe if we see such a small difference, this is maybe why we see these huge discrepancies between studies. So this experiment was high throughput, automated, but not reliable enough to do drug screening on it. So we tried to push it a bit and try to see, okay, we have some cytoplasmic TDP43, where does it localize? And recently it had been shown that TDP localizes into the mitochondria. So again, we tried to automate this analysis. So here we filtered the images. Again, the software recognizes itself the nucleus, the cytoplasm, and then here we have a TOM20 staining, and we get to get it to detect the TOM20 region. And here we can pull together TOM20 in orange, and in green you have the TDP bright spots in the nucleus. And so we can then quantify in an automated way the percentage of overlap between these two. And so, I'm missing some stars here. So here you see, again, we have 2 of our 3 lines that had an increased localization of TDP in the mitochondria, whereas this line we didn't see, it was the same levels as the controls. So again, here it was a parameter that was slightly increased, so we have higher localization of TDP in the mitochondria, but this was patient-specific, making it, again, a not reliable aspect for drug screening. So here I just showed you that we showed a slightly increased localization of TDP43 in the cytoplasm and in the near mitochondria in the TARDBP patients. I'm not going to show you, which was, again, not reliable, but I'm not going to show you here. We tried to get other parameters. We also saw increased neurite length and neurite branch points in these lines, but again, not a very easy parameter to follow. And we also saw that with aging of these motor neurons in vitro, we saw that we had an altered viability of specifically the TDP43 lines. But again, this was hard to follow. It means that we have to keep the neurons long and long in culture, meaning that we might lose them. So it gets a bit complicated to do drug screening on this stuff. So we thought that maybe what would be easier is to stress them and try to trigger the susceptibility that we see in the TDP43 lines. So this is what we put in place. We took a bunch of different drugs, which I know you must know all of them. We try to target ER stress, mitochondrial stress, inducing apoptosis, glutamate. And so we plated our motor neurons, matured them until day 24 because we still need to have mature motor neurons in there. And then we did a 24-hour, actually a bit longer, chronic treatment with all of these drugs at different concentrations. And with the Incusite, which is an automated, less complicated machine than the one that we saw before, we detected the neurite length. So this is an automated way of detecting the neurites without any dye in there. And then we also followed viability with a propidium iodide intake. And so we followed up all of these drugs, and we had interesting results only in sodium arsenite. I mean, this was a result where we saw that TARDBP lines were more susceptible to this stress than controls. So here I go in detail with the results. So here it was you have propidium iodide intake. So you have a control line here, you see the neurons. At 24 hours, you see that here some cells, they're starting to round up and start to die, but the PI intake is still not there. And at the same time point for the TDP lines, you see that almost half of the cells are already dead and PI intake is already in there. So here, compared in the dynamic assay, you can follow up for the 40 hours there. You can see that we have a premature death of TARDBP lines with low concentration of sodium arsenite. Eventually, the control lines still have cells that end up dying, but later and then less cells. And we saw the same actually when we looked at neurites. So after stress, you see that we saw the neurites kind of blebbing. And so the software didn't recognize anymore like very nice and straight neurites. And so we saw that with stress, we saw the premature degeneration of neurites of the control lines compared to of the TARDBP lines compared to the controls. So using this assay, we were able to kind of push the cells and get this nice difference that was quantifiable. So high throughput, automated, and this time reliable because first we have a lot of different points. So if you try to rescue, we have different time points that we can check. And so we decided to use this, and so we tested our favorite drug, riluzole. And very interesting, we were able to see here you have the TDP lines here and treated with different concentration of riluzole that go back to the control levels. However, riluzole in a very weird way, was also able to rescue the phenotype of the control cells. So it helped them endure better the stress, which it's oxidative stress, arousal. So why not? But so we are able to show that rizol, which doesn't really work very much in vitro usually, was able to work in this process. So I hope I was able to convince you that we still have work to improve the way we do drug screening, and we still can find better ways to do it. We, I think, are going in a direction where we are finding some interesting stuff. We still have to validate of what are we actually doing to the cells while stressing them with arsenite. So we launched a transcriptomics analysis to see how differently the controls and TDP lines respond to stress. And of course, we'll try a bunch of drugs on this. So yeah, I want to thank my lab, Sorana, my supervisor, and Manon, who helped me with creating so many neurons to do all of these experiments. And of course, I want to thank Institut Imagine and Medicia for giving me the opportunity to use these machines because without them, I couldn't have done any of this work. So yeah, thanks a lot.
Thank you, Elena, for this nice talk. Questions?
Hi, this is beautiful work.
Thank you.
I'm curious if you looked at TDP localization at this case. Because,
during stress?
Because maybe this is even a better readout. You know?
Actually, it is. So during stress, we see actually TDP. So the small difference that we saw, only after 6 hours of stress, so before they actually start dying, we see like this difference gets huge. Yeah. So this also becomes a new way of testing drugs here and then going back to check the TDP localization.
Any signs of aggregation or more localized in your image?
In the mitochondria. So when it goes out during stress in the cytoplasm, it even goes even more in the mitochondria.
And when you have some aggregation into the mitochondria, did you see some alterations of the mitochondria by electron microscopy, for example? Or?
No, we haven't checked that. We tried to look at it with a confocal microscope, and it's way too complicated because the cells are too small. But it will be interesting to see exactly what happens there.
Yeah, I was curious why the oligomycin didn't produce anything, given that we're all talking about mitochondria now. It blocks ATP production.
Yeah. Basically, I think there's also a game of concentration. For example, sodium arsenite, if you put it at a too high concentration, so above 200 micromolar, we don't see the same difference anymore. So it's really at low concentrations of arsenite that we see this difference. So maybe I missed the concentration of oligomycin where we could see this difference, because it seems to be very fragile there.
Does the colocalization with TOM20 change with the sodium arsenate?
It's increased. But yeah, it still goes there. Yeah, Some people will say it's not a clear localization in the mitochondria. I can only conclude that it's around there. I use TOM20, so it's an outer membrane marker. So for now, cannot convince you that it's inside of the mitochondria.
Thank you for the talk. I just have a quick question. Is your lab open to test drug candidates from, for example, biotech companies?
I don't know yet because our lines come from a hospital. I don't know how much we have the opportunity to use those for commercial issues. I don't know about that.
Thank you.
Thank you for your talk. On your last slide, it was the result for one mutation in particular in TARDBP. Do you have you reproduced your data for many mutations?
So I launched that experiment on Monday. I've not analyzed it yet, so I launched it on all of our lines. So I'm still waiting.
Okay, thank you.
But yeah, sorry, it was just on one line. Yeah.
Hi, here. Thank you for your talk. Maybe I missed the presentation because you have many iPS cell lines. Mm-hmm. What are the results with the CRISPR corrected isogenic lines? and also the one where you introduce the mutation. Do you have the same phenotype?
So basically, with the stress, I have the same phenotype. The only thing is that the COLF cell line seems to be more vulnerable to the stress. So basically, when I stress them, I see COLF, the control line, at the same levels as our TDP lines. And then where we introduce the two mutations, it goes even higher. So it's just that it's the same difference, but it's just shifted. So for now, I don't know really how to analyze this because it's just not the same response to stress. But in terms of neurite length and localization of TDP in the mitochondria and in the cytoplasm, it's really the same results in the isogenic and the patient lines, but in response to stress, it's just not the same.